Well, welcome back, students. Time for another lecture. This time on application A, which is divided into two parts. The first part talks about human hereditary disease at a very fundamental level, and the second part goes into depth as to what genetic counseling entails and the medical genetics health team. Some of you may end up in this particular field of medicine, and it's pretty important that you understand the definition of medical genetics before continuing. As the book says, medical genetics is a focus on diagnosis and management of medical, psychological, and social aspects of diseases involving mutations or influence of genes, i.e. the genetic basis of human disease. The membership of the health professional team can vary depending on the patient. If it's a child, it'll have one combination. If it's an adult, it may have a different combination. But more importantly, it depends on the disease itself, the genetic basis of that disease, and the environmental impact of that disease on the manifestation of the illness. Normally, once a healthcare team engages with a patient and their family, the interaction continues into the future, potentially affecting future offspring, unless, of course, the patient or their family decline continued interaction. At this point in time, all hereditary diseases of humans fall into three simple categories. The first is a single gene mutation, Mendelian fashion, that's inherited based on alleles at a single locus. The second type are multi-gene diseases, diseases involving more than one gene. And a mutation in any one of these can cause the hereditary disease. The third category is one of the most important and the ones that students really need to relate to, which is a combination of both genetic influences on the disease outcome as well as outside factors such as environmental and developmental circumstances. Regardless of the category of disease, the issue at stake is the correct diagnosis of the condition and then to propose answers and solutions in terms of medical treatment. With an expanding pool of genetic diseases being diagnosed, classified, and analyzed, the breakdown of these diseases can be again placed into three categories. The Mendelian conditions, where normally a single gene with multiple alleles is involved. Chromosomal conditions, where large chunks of DNA containing multiple genes can migrate between the genome. And these multifactorial conditions, where not just the genetics, but other environmental and developmental factors may be involved. One thing is for sure, that the individual disease, say, of a particular gene may not be identical in two different families because of these various parameters that may exist. One of the goals of a genetics course is to make sure that students have a rudimentary understanding of many aspects of genetics. And one of those comes inside the container called Mendelian conditions. When we discuss Mendelian conditions, and other chapters will be used to do that, we should leave with the sense that there are 6 patterns that can be observed in human genetic disease. The first are those diseases that follow the autosomal dominant mechanism of transmittance. Then the second is autosomal recessive. So these lie on the 22 autosomal chromosomes. Then we have the sex chromosomes. So diseases may be X-linked dominant and X-linked recessive. And because we only have one Y chromosome in most humans, then in this case, it's either going to be Y-linked or not. And finally, the one that students tend to forget is mitochondrial inheritance, because there are some diseases that migrate through mitochondrial inheritance. A second commodity of understanding for students is that mutations are the basis of variation in genes. So mutations give rise to alleles. But these mutations can be pre-existing and inherited from one or both parents, or they may manifest during the regeneration of the offspring. So they could be in the sperm or the eggs, or they could manifest at the zygote level and beyond. So those will be known as new mutations. They may not be present in the parents, but they will be in subsequent progeny. Thus, this complicates the medical genetics because the origination of the mutation may influence the risk of the condition in future offspring. With respect to chromosomal conditions, the manifestation of these may fall into these four simple buckets. So they may be due to the presence of an extra chromosome, a dosage effect, too much of that chromosome, not enough of the others. Remember, each chromosome can contain thousands of genes. So by having an extra chromosome, each one of those genes is now out of whack in terms of its ratio with the other genes on the other chromosomes. A chromosome can be missing. Now, when chromosomes are missing, that can have devastating effects. So this number 2 is less tolerated than number 1, but both are pretty devastating. Number 3 is where a small section of a chromosome may be duplicated or deleted. And the impact of that may be less dramatic than 1 and 2, but still significant depending on the size of the deletion or the duplication. And finally, certain structural rearrangements of chromosomes can take place. And this bucket here contains all kinds of mutations where an end of one chromosome may break off and be accidentally joined to the end of another chromosome. We are privy to the impact of these type of mutations, especially with the case of Down syndrome, where there's 3 copies of chromosome 21. Chromosome 21 is a tiny chromosome in the human genome, but it carries hundreds of genes, and those genes can upset that ratio that we spoke about a few minutes earlier. And these conditions may involve the autosomes or the sex chromosomes, and that relates back to the Mendelian conditions. The passage of chromosomal conditions is pretty rare because of their devastating effects on the individual. So it seems that these conditions are usually the result of new mutations in the child that were not present in the parents. Now, these changes to the chromosome number are often caused by errors during the cell division process known as meiosis. And that leads to a non-disjunction, a term that we'll pick up later in the course. Basically, this is an incorrect separation of the chromosomes during meiosis involving the cytoskeleton and the spindle fibers. Because these conditions are usually the result of spontaneous occurrence, their reoccurrence risk is normally low because the individual either doesn't produce any offspring or dies early before they have a chance to reproduce. Multifactorial conditions entail multiple factors, multiple conditions that can influence the interaction of multiple genes. And we're talking about environmental and developmental factors here. So as the book says, diabetes is an example. One thing to understand is that human populations have evolved in different geographical regions over many thousands and thousands of years, and that has led to different mutant alleles being present in different frequencies at different rates in different populations. So depending on the 2 parents, the frequency of these mutant alleles may be high, low, or somewhere in between. Now, the inherited genetic variation that comes along with the genetic information may make an individual more likely to develop a condition than others, but it all depends on both the environment and the genetics. Because these are mathematical estimates, we can use the word likelihood or probability to tell people what the chances of their future offspring are in developing these particular diseases. A medical jargon term, inherited susceptibility, is frequently used in conversations involving these likelihoods. Let's take a journey into a typical genetic intervention. It may begin with a couple planning a pregnancy. It may begin with a child being taken to the pediatrician, or it may begin with an adult visiting their doctor. Regardless, these personal physicians are the first to encounter the individual or family, but most physicians are unqualified or unprepared to take on the entire case. They may even not have the facilities locally, and therefore they may refer their patients to more professional and larger hospitals which have the resources to manage at a level necessary. Today, the reliance on specialist diagnosis has been overcome to some degree by the availability of direct-to-consumer testing kits. So we're talking about 23andMe, etc. albeit that these kits are limited in the number of genes and diseases they can test. The power of genetic testing still lies in the healthcare industry. These tests can fall into three main types. We have the molecular analysis tests, which look at DNA and RNA and look for particular mutations, making one susceptible to those conditions. And these are specific for Mendelian conditions. Then we have biochemical tests, which analyze blood samples and tissues, as well as the presence or absence of proteins, as well as their levels within the body. And they can test for variant proteins associated with a condition. And then we have those tests that look for chromosomal variation. And these are looking for changes in the arrangement or number of chromosomes, as well as unexplained mental impairment and physical abnormalities, which are normally associated with chromosomal mutations. And these can be at any age, not just in juveniles. The tests go beyond simply testing for molecular, biochemical, and physical rearrangements. The tests can also indicate other parameters. One such application is for carrier testing, where the test tries to identify individuals that are heterozygous for a recessive condition or have a chromosomal abnormality that could be then passed on to future offspring. Carriers are by definition unaffected by the disease itself. The term carrier indicates that one of their chromosomes is carrying a recessive version of a gene that can then be passed on potentially to future offspring. Unfortunately, because of the historical legacy of human populations, some groups have large numbers of carriers. One example is the Ashkenazi Jewish populations of New York, which were founded by a small group of settlers that then continued to interbreed amongst themselves. This group is famous in genetic circles for the presence of an incurable disease known as Tay-Sachs. And we'll talk about Tay-Sachs disease at another time. For now, in this context, a large number of members of this population are carriers. And if they interbreed, then because this is a recessive disease, 2 copies of these alleles may manifest in the children at a very high frequency. Other forms of testing available are pre-symptomatic testing. In this case, the patient seeks genetic testing to diagnose for the future. Examples of this include diseases which are late onset. Late onset simply means that the disease does not show itself until later in life. And the classic example of that is Huntington disease. In Huntington disease, people normally start exhibiting the symptoms around age 40. However, half of the people don't exhibit anything until much later in life. The issue is that by this age, most of the patients have already had families, so they have already passed on these alleles to their children. This then raises ethical and moral questions when an individual gets tested, such as who needs to know, when they need to know, why do they need to know. Other tests focus on the newborn, and in this particular case, this application has more or less been adopted by every state in the Union and many countries beyond. So these are mandated tests. Now it's important to understand that not every state has the same number of mandated tests. So some states have more, others have less. Therefore, potential parents should check with their state to see which tests are available. At this time, in certain states, the panel of tests is approaching 50 in number. What are these panels of tests testing for? And the answer is rare genetic diseases that have been shown to be either preventable or greatly reduced in their effect by early biochemical or medical intervention. The final category is prenatal testing, which is administered sometime during pregnancy at specified periods. And these can allow the scientists to detect chromosomal or genetic conditions. These are performed through 3 routine procedures, 2 of which are quite old. The first is chorionic villus sampling, which takes a small section of tissue from the placenta. And the second is amniotic fluid sampling, which is done through a needle embedded into the amniotic fluid. The third category, which is quite new, is simply taking mother's blood and testing for certain markers that are released from the pregnancy into the circulation of the mother. The second half of the chapter deals with genetic counseling and its interactions with both the patient side and then the medical team side. Genetic counselor is not just an individual who understands the genetic basis of disease and can calculate the probability of that particular disease being manifest in that particular family. They are also individuals who are trained as counselors. During the process of interaction, they can talk about the upcoming genetic tests that they believe should be available to that family. And once the tests reveal the results, then they can counsel and guide their patients towards an outcome that's appropriate for that particular family. So they don't tell the family what to do, they give advice and guidance so the family can choose for themselves what the best options are. Incidentally, the name applied to the patient is the consultant. So the consultant liaises with the consultant in the family. Genetic counselors are also required to communicate their findings and their suggestions to the medical team that's intervening in that particular case. Genetic counseling is thus an ongoing process of communication that initiates once the patient has been accepted and continues well into the future. Ongoing interaction between the family and the medical team. Table A1 outlines some of the common indicators for a personal physician, pediatrician, or a general practitioner to alert or suggest genetic counseling. Table A2 outlines some of the goals of genetic counseling pertaining to the family. An important skill possessed by genetic counselors is the ability to communicate in a comprehensive yet understandable manner medical information, both about the current condition and the reoccurrence of that condition as a risk into the future. That entails communicating mathematical concepts so that the consultant is able to make immediate decisions as well as long-term decisions pertaining to health matters. Over the next few slides, we're going to look at how the assessment of future risk is calculated using mathematical and pedigree analysis. Students are not expected to know in depth how to calculate these, but have a rudimentary idea of where the principles come from. In most counseling interventions, one is looking to estimate the likelihood of a future pregnancy in terms of a fetus having a particular genetic condition. These estimates are complicated by the inheritance pattern of that particular trait, the structure of the family, and the information available about the family members for that analysis. In most cases, it's suffice to say that the surest way to determine these probabilities is to test directly for the genotype. But unfortunately, in a future pregnancy, the fetus has not yet been conceived, so that is an impossible task. The alternative is the probability estimation technique that we'll talk about right now. There are certain mathematical and statistical processes that are way beyond the scope of this chapter. Students are not required to learn the exact process, but are asked to pay attention to the logic employed in the background. One such probability estimation methodology was developed by Reverend Thomas Bayes and is known as Bayesian analysis. And it's simply a method that geneticists have adopted to supplement Mendelian inheritance patterns and ratios. The goal is to use as much information about the family tree, the pedigree, as possible in more accurately relaying the risk of a particular future individual being born with a genetic disease. Bayesian analysis is broken into 3 steps. 2 of the steps are easy to understand. The last one is simply multiplying the answer to this with the answer to that. The second easier one to understand is step 2, where one is simply going to look at other babies produced by the same parents as the potential baby that they will have in the future. So you're looking at brothers and sisters in this case. And the first part of the analysis looks at prior probability of the genotype in the grandparents and uncles and aunts and uses that to then estimate a probability. The first example given is to do with this pedigree shown here. So we have 3 generations, 1, 2, and 3. And we have 2 individuals in the first generation, individual 1 and 2. We have 2 individuals in the second generation, 2, 1 and 2, 2. And then we have the future child indicated by a diamond because its sex has not yet been conceived, and that's going to be 3, 1. The question being asked is, what is the likelihood that 3, 1 will have a disease? A rule in pedigree analysis is if somebody is marrying into the family, so 2, 2, a female, indicated with the circle, is marrying this male, and they are going to be having the child, the pedigree of this individual may or may not be known. But in this case, it doesn't concern us directly, so we can assume that the presence of that condition in this individual is at the same frequency, the same level as in the population as a whole. So in this example, we are told that the frequency of carrying an allele in the heterozygous condition for an autosomal recessive disease for this population is 4%. And that number can vary depending on the disease and the population. In the pedigree shown, the grandmother of the potential child had the disease. So the grandmother must be small a, homozygous recessive. Again, because this is an autosomal recessive disease, the only way this individual can have the disease, indicated by a filled circle, is if they have 2 mutant alleles. Grandfather here could be either homozygous dominant, big A, or he could be homozygous recessive, big A, small a. Regardless, the big A prevents him from manifesting the disease. So the question is, which combination of chromosomes ended up in their son in generation 2, 1? Because of Mendelian inheritance, we know that this son must have got one of the 2 small a's from his mother. So he's at least going to be 1 small a. And because he doesn't have the disease, we don't have a shaded box, he must have got the big A from his father. So he must be heterozygous. So these facts are known, especially for an autosomal recessive disease. Combining this knowledge with the probability that his spouse has the allele at 4%, we can then estimate the probability of their future child having the homozygous recessive condition, and thus the disease. On this slide, we've annotated it with the information that we just spoke about on the previous slide. So 2, 2 is coming into the marriage at a heterozygous frequency of 0.04. 0.04 is 4%, written in a relative manner. Then 2, 1, the son, has the heterozygous condition for the reason we just explained. We know that for sure. With this knowledge in hand, we can now calculate the probability of the offspring, the future offspring, having that condition. If both parents are heterozygous, then there will be a 1/4 probability that this offspring, because of the union of sperm and egg in that random fashion, will have the small a genotype. So 1/4 is 0.25. So 1/4 of the offspring that they have, 1 child in 4 will have the condition. We can take that knowledge, the certainty about the male, and the probability from the population about the female, and multiply them together. So the female brings in another parameter, and that parameter is the frequency of the heterozygous condition in the population. So we multiply that by the 1/4 probability that a heterozygous and a heterozygous individual will produce a homozygous recessive child. And once you do the mathematics, you come up with this answer here. So for this, the revised probability of that child, based on the evidence coming from the grandparents, is 1% that they will have the condition. The second example is very similar. The only difference is that we have more information coming from the pedigree than in the first example. So once again, we have a couple that want to have a future child, and they want to know what is the probability based on this pedigree on the mother's side of that child having the genetic disease. Breaking the problem down into the three parts that we mentioned earlier, the first part was the prior probability. The prior probability simply looks at the mother and her pedigree and the way that individuals in either the same generation or previous generations have manifest the disease. And that will then alter our understanding of the chances of her inheriting the mutant allele. Once we've done that, then we'll look at the conditional probability based on her and him uniting to form an offspring. And then we'll just multiply the two together to find the answer for the baby itself. The important thing to note in the second example here is that this is not an autosomal recessive disease. This is now an X-linked recessive disease. X-linked. That complicates the analysis slightly, but it also makes certain features easier to understand. So let's look at that. When genes are located on the X chromosome, we have to understand that females have 2 copies of the X chromosome and males have 1. As important as that knowledge is the knowledge that males do not get their X chromosome from their father. They do not get their X chromosome from their father. They always get their X chromosome from their mother. So they get an X from the mother and a Y from the father, and the XY makes that individual male. But that's a different story. So we can use that information to calculate the risk for the parents of the potential mother, the grandparents here. Focusing our attention on the grandmother, she has 2 brothers, and those brothers have the disease. The fact that they have the disease means that the chromosome must have come down from the great-grandparents. So the mother here has 2 X chromosomes, and the father here will have 1 X chromosome. Now, if he had the disease, there's no counterpart to the X chromosome, and therefore he should be shaded. So we know for sure that his X chromosome is normal. The fact that she doesn't have the disease tells us that one of her X chromosomes is normal, but the other X chromosome must be mutant. So she's heterozygous and he's hemizygous. Hemi means only 1 copy of a chromosome. The other Y doesn't make any difference. So this son here was unlucky to get the mutant chromosome. She would have either got the mutant chromosome or the normal chromosome from the mother, but she's definitely got the normal chromosome from the father. Because if she got the Y chromosome, then that would make her a male, and therefore that would be a box, not a circle. So she got the normal chromosome from her father, but which of the 2 chromosomes did she get from her mother? The normal one or the mutant one? We're not sure. There's not enough information in the pedigree to deduce that. And that's the problem. That problem cannot be alleviated because the pedigree is so small. Regardless, we do know from the way that genes are inherited in the Mendelian fashion that the 2-2 individual here, this female, the grandmother, has a 50% chance that she carries the normal chromosome and 50% chance that she carries the mutant chromosome from her mother. One way to tell her condition is to look at her children, especially her sons, because she would be passing to her sons 1 of her X chromosomes. Therefore, each son will only have 1 X chromosome. If they get the one that's normal, they will be normal. If they get the one that's mutant, they will have the disease. So we can see here that 3 sons have been born, each one with a 50-50 chance of getting either the normal X or the mutant X. So what we do, we take the probability of each son, and there's 3 sons total, and we multiply them together. So 1/2 for this son, 1/2 for that son, and 1/2 for that son. So that gives us a ratio of 1/8. And we can make use of that mathematics in then multiplying this value here by that, as has been done here. So this is the prior probability, this is the conditional probability, and the sum of these two will tell us the probability of this individual being heterozygous. We now have to look at the other side of the coin. And the other side of the coin is that the grandmother is not a carrier. So in the previous example, we looked at her being a carrier. Now we're going to look at her not being a carrier. So in this regard, the prior probability that she is not a carrier is 50%. Again, based on the mathematics of Darwinian inheritance. Now, given that 2,2 is not a carrier, the conditional probability that she passes a dominant bearing X to her child is 100%. There's no way that she can pass to any of these children a mutant form of the allele. Therefore, she will always pass on the normal form. And when something is 100% certain, we give it a probability of 1. So the joint probability here is going to be 1/2 that she inherited from her parents, the mutant allele, times 1 that if she doesn't have the mutant allele, that her daughter will get the mutant allele. And we multiply them two together and we get 1/2. So that's the joint probability that she will inherit the mutant allele. If one were to follow through with the entire analysis, which you are not required to do so, you will end up with a value. And this is the value that their child will have the likelihood of having the condition if it was conceived based on this pedigree. It's not 25%. Because of all this other influence of this pedigree, we can now conclude that the probability of them having a child with the condition is just 1.4%. The mathematics behind the individual components, the two hypotheses, that she is a carrier and that she's not a carrier, are indicated here in this figure. And this pertains to the grandmother. Students are not required to learn the overall mathematics, just the logic behind each modification of Mendelian inheritance. The last part of the chapter addresses the ethical issues coming out of the field of medical genetics. One thing for sure, the speed at which technology has enabled us to decipher genetic diseases has outpaced public debate over ethical guidelines and the laws that manifest from them. Indeed, this dilemma was foreseen before the Human Genome Project began, because a substantial amount of money was set aside for addressing the concerns pertaining to ethical, legal, and social implications of those research findings. The goal of the ELSI project was to concurrently develop these guidelines as knowledge was beginning to emerge from the project itself. Two relatively recent laws have reinforced protections for patients' personal genetic information. In the case of the Affordable Care Act, or Obamacare, this was part of the pre-existing conditions clause that was part of that law. The ethical standards that have evolved from the debate have at their core 3 principles that guide the use of genetic information in medicine. The first being the likely benefit of medical genetics to the community. Second, the respect for individual autonomy in making decisions about one's own healthcare decisions. Number 3, the preservation of fairness and equitable treatment for all individuals regardless of genetic findings. At this time and into the foreseeable future, our technology will not let us address chromosomal conditions because of the degree of genetic information involved. With no treatment options, it's likely that the choice for the parents will be either to continue or terminate these pregnancies. In the current political climate, with challenges being made to abortion rights, these choices may not be for the parents in the future. Certain interventions, such as newborn genetic screening, have not raised the same level of public debate, whereby these procedures are commonly accepted by many, if not all. The ethical and moral debates around genetic predisposition testing are still in the air. It may come down to a personal choice for an individual or a family to decide on which action to take. The text talks about Huntington disease and its late age of onset, and the fact that conceptions of offspring have already taken place. What happens in that case if one individual wishes to know and the other individual does not? Indeed, it goes even deeper. In some cases, the presence of mutations has led to individuals taking precautionary actions just in case their bodies develop disease, as was the case with Angelina Jolie, where she had both her breasts and her ovaries removed. Each one of us is part of this ongoing debate. One thing for sure, the more knowledge you have, the better your contribution to the final outcome.